Welcome to video lesson 1 on the antigenic diversity of the influenza virus. The CDC and NIH both recommend receiving an annual vaccine for the flu. Flu vaccines contain 3 or 4 of the most common A and B type strains of the flu for that year. Vaccines prime host immune systems to recognize and respond to invasive microbes by using mild immune responses to produce antibodies. Antibodies induced by vaccination allow the body to remember a disease and respond more swiftly to invasion than if the vaccine had not been applied. Vaccines are highly effective against fairly stable diseases such as measles, chickenpox, and polio without needing yearly vaccine shot updates. So, why do we need an annual vaccine for the flu? The structure of the flu genome for A and B viruses consists of 8 RNA strands, each of which codes for functionally important proteins, such as the hemagglutinin and neuraminidase proteins. Hemagglutinin and neuraminidase are the key antigenic proteins used by the flu virus to infiltrate cells. Neuraminidase later assists newly produced viruses with escaping the host cell. Neuraminidase inhibition prevents the virus from escaping and spreading to other cells. Scientists refer to these proteins as NA and HA, as will I for the rest of the video. There are 16 subtypes of HA and 9 subtypes of NA. From the subtype combinations, scientists name flu strains. For example, H1N1 is composed of HA subtype 1 and NA subtype 1. Without a vaccine to provide antibody recognition of a specific subtype, and in the absence of natural immunity, HA proteins bind to external sugars and sialic acid sites on cell walls. The bound cell is tricked into endocytosing the virus, and replication within the host cell begins. During the process of replication, mutation or reassortment may occur in the genome of the virus that results in changes in the expression of antigens. This process is called antigenic drift and is the primary reason for needing a yearly vaccine, as well as explaining how people can get the flu more than once. Antigenic drift is a series of very small changes that happen slowly, with reasonably subtle results as the virus moves from host to host. Once enough differences in the expression of the virus and the expression of the viral proteins have accumulated, the body no longer recognizes the flu virus and it may invade successfully. Antigenic shift is a more abrupt process. In antigenic shift, type A flu viruses undergo major change in their antigenic proteins, allowing the strain to jump between host species. For example, the H1N1 pandemic of 2009 was the result of an antigenic shift, allowing that strain to move from pigs to humans. There is typically very low immunity to novel viruses after antigenic shift. The CDC recommends that everyone 6 months and older be vaccinated against the flu every year, especially to provide herd immunity to those who are immunocompromised and/or cannot receive the flu vaccine for health reasons. Because not enough people receive their vaccinations, between 5 and 20% of the United States population is infected with the flu every year, with more than 200,000 people needing hospitalization. Deaths have ranged from a low of 3,000 flu-related fatalities to a high of 49,000 over the past 30 years, from 1976 to 2006. In other words, get your flu shot. Please review these lesson objectives.